Our next speaker is Richard Durbin.
Okay, so thank you for inviting me to speak. So, we heard a couple of days ago about the 100th anniversary of the journal Genetics and of James Crow. But it's also the 100th anniversary of somebody else in our field, who's Francis Crick. Actually, we celebrated his birthday in Cambridge. The local brewery brewed a beer, the Molecule of Life, to celebrate his 100th birthday of the co-discoverer of the structure of DNA. And I think that's relevant because, well, James Crow, in fact, referred to the 20th century as the century of genetics. The ability to sequence DNA, I think, and look at genomes, as we've heard in the first two talks, is really going to mean, I suspect, that the next century is going to be even more the century of genetics going forwards. Our ability to do everything directly observing the substrate of genetics. And in that light, I'd like to argue we're still in the era of genome sequencing. People tend to think that the first era of getting reference genomes 10 or 15 years ago happened, and then population sequencing with the various 1001, 1002 genomes projects. But now we can really, as we've heard, in fact, in the first two talks, start to sequence the genomes of essentially any organism and look at diversity within them and look at the genetic processes that are going on to understand what's happening. And genome sequence is the product of evolution, and I think we also heard this yesterday in the overall keynotes. Each of those referred to population genetic arguments, even the broader subject of genetics, reveal what's going on in terms of the genome. So I'm going to talk about Lake Malawi cichlids, which are perhaps the most dramatic evolutionary radiation that we know about in vertebrates. And there are hundreds of different species in the East African Great Lakes. Cichlids also have diversified dramatically in other places in the world, but particularly in lakes Victoria and Tanganyika and Malawi in East Africa. There are very big sets of species with enormously different morphologies and ecologies and having filled a huge variety of different niches. And in Malawi, this is believed to have happened within the last million years or so, and there are some number, 500 or to 1,000 species, depending upon how they've been counted. So there have been people who've worked on this for a long time, but we're just now beginning to be able to look at the genomes which underlie that. And we'd like to know what the genetic structure of the differences are, how are they related, what's going on in adaptation and speciation, and also what the genetic basis of the differentiated traits are, what are the selected genes and mechanisms. So in collaboration with George Turner and Eric Miska and a number of other colleagues, we started doing this at the Sanger Institute. And I'm going to tell you about the initial stages of this. It's a little like Andy Clark's talk about his system, where really this is just opening up, I think, a whole set of opportunities. So we sequenced 80 species from Lake Malawi with essentially one per species. There's a number of low-coverage samples on top of that. And then in an initial study in a crater lake nearby, there are 50 samples of each of two different morphs. Shown here, there's a small yellow morph and a blue morph, essentially much more recently diverged. So, because this is a crater lake that was formed in the last few tens of thousands of years by a volcanic explosion, essentially. And those were sequenced at low coverage, and I'm going to tell you briefly about those on the next slide. The genome size is about just under a gigabase. There's a reference genome that was obtained by Federico De Palma and colleagues at the Broad Institute, and in fact, one for each of the Great Lakes and for now tilapia. And the heterozygosity is about 1 in 1,000, comparable to human and many other species. vertebrate species. So the Crater Lake story, this is a picture. It's just 800 meters across. It's one of a set of lakes. There's a little picture here. You can see these little round blue dots. They're all small crater lakes. They've each got populations of this Tilapia calliptera, which is the nearby riverine generalist fish, and there are separated forms occurring in more than one of these lakes. So potentially there's the opportunity for replication experiments there. So this was published at the end of last year by Milan Malinsky, student in my group, and others. So I'm going to summarize it very briefly. Essentially, these are the ones from Lake Massoko, from the Crater Lake. This is the nearby river. This is another lake, Itamba, and outgroups. So it looks to be a sort of monophyletic group, or in some sense, maybe that's not quite the right word, but a singly separated from the others. And the blue form, which is the more derived form, the yellow form is found around the shore of the lake. The shallower water, the blue form is benthic, has a different head shape and a whole variety of different characteristics. There's also different diet, appears to be, sub-monophyletic from within that particular, from within the lake population. On a PCA plot, the blue separates from the yellow with the first principal component, with 3% of the total genetic variation. The gray dots are small forms which are probably not mature yet. And overall, the FST between these two populations is only 3%, but if we look genome-wide, we see these positions. There's nothing that's fixed. There's no fixed differences, but we see high peaks of FST that are clustered on the genome but are separated by baseline levels of diversity in this sort of structure here. And there are 50 to 100 of these islands with strong signals both in FST and DXY, most of which are around genes where there's no coding difference at all. But interestingly, rhodopsin, which you might imagine the visual system is quite different in deep and shallow water, there are 4 non-synonymous changes, and we showed that there was actually a frequency change there. So moving on to the whole lake, we can build a whole genome phylogeny. There are very long tips, which are in fact individual samples. Some genera are not monophyletic. There's a whole sort of interspersed set of genera here, which really, where the classical phylogeny or taxonomy was not consistent. Some well-known forms, taxonomic groups have separated out. But there's lots of repeated specializations with respect to the tree in head shape and diet and coloring and so on. You can see here some of the big variety of shapes. This is a pelagic form that's like a sort of piscivore, and then these crunch snails, and there are ones that specialize in eating scales of other fish and all sorts of specializations. If we look at FST, though, there's segregating variation across the entire phylogeny. So heterozygosity, as I said, is between about half and 1 in 1,000, but the divergence is less than 1% between any pair of these species. And so FST between species varies between 5% and 65%. And if you compare that, that's, the FST within zebrafish actually is comparable to across the entire, within Danioreria, across the entire radiation. And so you can also cross these in the lab or by in vitro fertilization if they're really extremely different. So there are opportunities for crosses and for potentially association genetics across the system. So what about the other sort of large-scale properties? We've estimated the mutation rate. It looks as though the radiation is even younger than 1 million years, maybe around 500,000 years. Because there's this segregating variation, actually the tree, there's a consensus tree, but if you cut the genome into windows, actually if you make 1,500 windows that are, I think, 500 variants wide, each of those, the trees in those is different. There's no one tree that recurs. So there's a sort of consensus form here. So that could arise either through incomplete lineage sorting or through hybridization. And obviously there's a really interesting question as to whether there's been hybridization involved in, and gene flow involved in the speciation and separation, segregation of traits. So we looked at that using Abba-Baba type tests. We heard briefly about these. So these were introduced actually in the context of human genetics, asking most famously whether there was a Neanderthal component in the out-of-Africa humans. And what happened there is you have an outgroup and then a potential sort of group here, the Neanderthal, from which you might expect, you might be looking to test whether there's some gene flow between the Neanderthal and one of the two modern human groups. And if there was even with ILS and segregating variation, if there was a gene flow between, well, if there was no gene flow, then you'd expect a symmetric relationship. These two would be symmetric with respect to this group here. So the test is quite simple. You look for variants which are either ABBA or BABA, and those should be symmetric, and so the difference between the numbers of these and the numbers of these should be 0 under the null hypothesis. And you can test for deviation from the null hypothesis, and in this case, there's a 2 to 3% additional Neanderthal-like component in the Europeans compared to the Africans. So if we do this we've done this, in fact, all three-way D-statistics on the major Lake Malawi lineages using a Tanganyika sequence as the outgroup. And this is shown here with this sort of the x-axis as the x group and the y-axis this way, and the inset is the name of the inset. And what you can see is there's some big bubbles here, and the maximum here is 41%. So that's nearly, essentially, a complete removal of tree structure. So there's enormous signals for gene flow. And in particular, we can see in red, there's evidence for gene flow within Lake Malawi compared to an in-group river population. So that's where these are both, that's a riverine population that ought to be more like this, but actually these things in Lake Malawi are more like other Lake Malawi fish, so there's a gene flow in Lake Malawi. There's gene flow shown here in green for differential gene flow within Lake Malawi between groups that are living nearer the shore compared to further out. And then on the larger scale, around the edges of this, there's evidence for a complex history of East African cichlid radiation and gene flow between the Great Lakes and the riverine fish in the whole of East Africa. So on the large scale, there's evidence that there isn't a simple sort of separation of species. Further evidence for gene flow is shown here. This is a PCA plot of the various species, Malawi species. And what you see here is a sort of cline. Now, if you have complete tree-like phylogeny of species, you'd expect there to be clusters. You see in this sort of cline, for example, with admixture between human populations. But in this case, these are different species, and furthermore, the ones at this end are all shallow water, and these are mid-depth, and these are deep water, so it suggests there's been gene flow across different depths of populations. And how am I doing for time? But if we go to the sort of fine scale, so this is all at the coarse scale, if we start going to the fine scale, interestingly, this is a genus where phylogenetically you might think there'd be evidence for gene flow. We see very little. So maybe there was evidence for a lot of gene flow early on in the radiation, but not so much genome-wide in the recent separation of these species. So here, this was a sort of strong candidate, we thought. There's only 0.37% and a non-significant Z-score. That's genome-wide, but on a local scale, we find things that are beyond 4 standard deviations away from the mean, and so there's some evidence here, possibly, of separations, but we're not sure. We need bigger numbers to look for that. We also have started to look for pairwise comparisons of species using multiple samples, as we did within the crater lake, but again, the numbers are small here. But there are, again, it looks like here the mean FST is about just under 10%, but there appear to be these spikes, and that again is suspicious. So if we look overall across the whole radiation, this is looking at an excess of non-synonymous versus non-synonymous segregating variants on the x-axis, and looking at a relative increase in differential allele frequency for non-synonymous compared to synonymous variants. Then there appears to be a concentration of genes involved in vision and in the immune system, interestingly, in the sort of upper right section where those signals will be most strong. So here's a whole set of opsins, of things involved in photoreceptor recovery time and function, and other photoreceptor genes. So we're collecting more fish. I think there's enormous opportunities here, both to observe directly and, in fact, to set up crosses and do QTL studies. We've now got fish from over 240 species and larger numbers so that we can get better numbers for these things. We're making data available. We're trying to really empower a whole set of studies. And I want to thank Milan and Hannes, two outstanding. Well, Milan is now a postdoc, and Hannes is a postdoc who came from Magnus Norberg's group. And in particular, George Turner and Martin and their colleagues who've worked on cichlids for decades and worked on the ecology and the evolution. And it's really marrying that with the genomics, I think, is going to provide enormous opportunities going forwards.